Today's lesson is on the DNA structure. We've been studying a little bit about DNA now. Now we're going to take a look at deconstructing it, looking at all the pieces and parts, and understanding how it works. It's an elegant structure. In 1953, Watson and Crick described it in a paper. And that paper was literally about a page and a half. It was very elegant and to the point, just like the design that they were working with. So let me read just a sentence, an opening sentence from it. It has not escaped our notice that the pairing that we have postulated immediately suggests a possible copying mechanism for the genetic material. So it started off a relatively simple, with a very simple description, and of course, as they went through it, this particular structure is not only elegant, but also very, very simple. It's been said that the humans are made up of about 6 billion different base pairs to make us human, whereas a chimp's, our closest relative, has only 98% of that, while a banana has 97%. So that tells you the power of just what a little change can make in the DNA structure. So let's go ahead and get started on some of the basics. As you can see in this picture that I have before you, it really is all about the double helix. And most everybody knows or has heard about a double helix. It's basically a ladder that's been kind of twisted, and it looks like a spiral staircase. This double helix, of course, coils around itself, and just like a spiral staircase, it wraps around and around. And in between those handrails, so to speak, we also have what we call base pairs, and we'll get to that in a minute. But just for now, you should know that it definitely is a double helix. Now, on the next slide, we're going to go through what the basic unit of structure is, the backbone, if you will. Again, as you can see here, it has that double helix, and if I were to draw a line, it kind of goes in this direction, just like this. And then at one end, it has what we call like a prime number, like 3 prime. And we'll talk more about that. On the other side is the 5 prime end. Like I said, we'll talk about that a little bit later on. Now, on the other direction, though, on this other strand, it goes in the opposite direction. And it, too, will have a 3 prime. And then over here, a 5 prime. Even though they're very parallel, they're going to be actually adding to the length in a different direction. And that's what we call anti-parallel. So the basic structure is a double helix, but it's anti-parallel because it's built in opposite direction. It flows in opposite direction. And the backbone of that, what puts all this together, is what we call a nucleotide. And this piece right here, these are all nucleotides. 1, 2, 3 nucleotides. They're made up of a particular structure, and that nucleotide has 3 distinct parts. It has a base, a nitrogenous base, and there are 4 different kinds of them, by the way. It also has a sugar on it, and in this case, it's deoxyribose. And it's deoxyribose. And that is a different kind of a sugar because when we look at RNA, it's going to be ribose. And then the next piece besides the base, we have the sugar, and then we have the phosphate group. These are all the 3 different parts. And of course, if we looked at another one here, you can see that here is another one. That right there is the sugar, deoxyribose. This is the base. And this over here is the phosphate. Every nucleotide, phosphate. Every nucleotide is made up of those 3 things: a phosphate, a sugar, and a base. Base, sugar, phosphate. So those components are part of every DNA molecule. And then there are the bases. There are 4 different kinds of bases. There are 4 bases, and they're located here, here, let's see, here, and there. And those 4 bases are very specific to how they pair with one another. For instance, if we take a look at the different classes, there are basically 2 classes of bases. Number 1 is a purine. I'm not going to go into detail on their structure this particular time. At a later one that I'll be able to do that. The other one is what we call pyrimidine. Oops, let's try that again. Purine is one type. Number 2 is pyrimidine. So each one of those 4 are classified as either a purine or a pyrimidine. For the purines, we have guanine, and we also have adenine. And in this particular depiction, the blue right here is guanine, so I'll just put a G on it. And then, of course, adenine is green, and I'm going to put a, let's see here, let's change colors here real quick. And green will be adenine. I'm sorry, red will be adenine. So the green one is, excuse me, the blue dot is guanine, and the red ball or dot is adenine. And then when we get to the pyrimidines, we're going to have cysteine, and they're also going to have thymine. Thymine in this picture is going to be green. Put that with a T. And then the other thing, guanine will be orange in this picture, or cysteine is going to be the orange ball. So in this particular case, as you can see here, we always have A is going to bond with T, and C is going to bond with G. So adenine and thymine are going to bond, and then cysteine and guanine are going to bond, or they can go in reverse order. But basically, in any case in DNA, A and T go together, C and G go together, and of course they can be reversed. But we just can't have the crossover. It's guanine and cysteine, and adenine and thymine. That's just the way it kind of makes the bases fit together. So that is on bases. Now we need to move on to bonds. There's 2 different kinds of bonds, but 2 of them are very similar in the way that they work. The first kind of bond that we have is what we call a glycosine bond. So glycosine. I should say glycosidic. And basically what that is between a sugar and a base. So that is the glycosidic bond. The second one is called a phosphodiester bond. I'll just call it phospho. And what that does is a sugar to phosphate. So when we look at a glycosidic bond, it's the base versus the sugar, and this is the sugar, and it's going to be this bond right here. When we look at the phosphodiester bond, it's going to be between the sugar and the phosphate right here. So those are 2 bonds that are very, very kind of rigid. They hang on to things extremely tightly, doesn't let go, and that makes a lot of sense because it's the backbone of our DNA molecule. But the one that needs to be a little less aggressive would be our third one, and that's our hydrogen bond. And our hydrogen bond is this blue one. And notice that it's kind of thinner and lighter, and it's not quite as stout as the others. So this one is the one that's easy break. And again, that makes sense because when this divides and copies itself, it's going to break right down the hydrogen bonds. So that are the 3 bonds. Now the other part that's kind of fascinating to me is the ends. As it begins to divide, when it starts to replicate itself, it unzips. And it unzips right along this plane right here. And then if you follow the hydrogen bonds, that's the plane it follows. It just unzips it. And as it unzips it, it's going to expose all of those unbounded bases. Because now those bases are open for free bases that are in the nucleus to come along and find the mate and then lock in tight. And when that happens, though, it happens in a particular way. As the chain is added to or lengthened, it only happens in one specific kind of way. In this particular diagram right here, you'll notice that this magenta and then this yellow. This magenta is what we call the 3' end. The yellow is the 5' end. Now again, this strand right here is an entirely complete different strand than the one that the 5' end is on. So if we were to follow this, it goes like, it goes like this. And the other one, change colors, comes around like that. So those are 2 different strands. Each one has a 3', each has a 5' end. And you might want to ask, well, why is that important? Really because the next nucleotide on this end is going to be added here, and then here, and then here. So it's going to grow or lengthen on this end, on this 3' end only. Doesn't do it on the 5'. And one of the reasons is it makes it go a lot faster when it's adding or copying. Whereas if they were competing for basically the same sites, you might kind of a logjam might slow things down. But on the other end is where that one lengthens. So it's lengthening itself at the 2 separate ends, so it kind of goes apart and they don't get in the way of each other. So those are the ends. Now the way it looks, let me see what I can do to draw a sugar molecule, or its conformation, I should say. On one end, you're going to have a base. I'll just depict that by a B. And another end here, you're going to have a phosphate. I'll draw a P there. And then what you're going to do is on this base end over here, you're going to have 1', 2', 3', 4', and then right here is 5'. This denotes the location of all the carbons. This is really carbon-heavy molecule, and this is called the 5', and this is called the 3'. So on this strand, which is a 3', this carbon is kind of sticking out right there, this corner sticking out. On the 5', the phosphate blocks any kind of bonding that's going on, and that would be right here. So the ends are 3 and 5'. So that should give you more of an indication of how it's going to put together. And by the way, because of this 3' and 5', even though they're parallel to each other, they lengthen in opposite directions. So that would be called an anti-parallel strand. Even though it's parallel, one is going in one direction and the other is going in the other direction. So that would be anti-parallel. So just kind of in brief review, the actual structure is a double helix. It's made up of 4 different bases that go together a certain way. Adenine goes with thymine. Cytosine goes with guanine. They also broke up in purines and pyrimidines. And there are 3 different kinds of bonds. And they are glycosidic. And also it's phosphodieesters. And of course, those make up the backbone to make it really rigid and strong, whereas the hydrogen bonds are a little bit weaker than that, and they give away easier so they can be copied. And then we have ends that are called 3' and 5', and the lengths are added at 3' away. I hope this helps describe it well for you, so that as you are putting together your models and you're labeling them and then you're writing basically a paragraph on how the structure goes together, this will help you a lot.